everybody, it's Mrs. Pound, and we are studying Chapter 7 on Biotechnology from BJU Biology's 4th edition. We're currently studying Section 7A on Biotechnology, and today we're going to focus on subsection 7.3, Stem Cell Technology. So our objectives for today will be to define stem cell and explain the difference between somatic and embryonic stem cells. So what is a stem cell? A stem cell is any cell that can divide to differentiate into specialized cells. So it is just a general cell that is not specialized yet, is not, say, a blood cell or a nerve cell, a neuron, or anything like that, but it could become any of those. Stem cells can be described as totipotent. If it's totipotent, it means that just after fertilization, a single-cell zygote has the potential to give rise to all of the cell types in a human body. So it's like we're saying it's totally potent. It can totally become anything. After the zygote develops and the embryo moves further along, the stem cells become pluripotent. This means that stem cells can give rise to many, but not all cell types. So it has become slightly differentiated, but it's still a stem cell. So sources of stem cells. There are two main sources where we get them. There are embryonic stem cells. This is an undifferentiated cell taken from an embryo that has the potential to give rise to various other cell or tissue types. Now, when these are harvested, though, the embryo is destroyed. So, Genesis 9:6, which states, "Whosoever sheddeth man's blood, by man shall his blood be shed," could be used to oppose the use of embryonic stem cells. We can also obtain stem cells that are somatic. Somatic stem cells are obtained from the differentiated body tissue of adults and children. You actually have many stem cells throughout your body, and probably one that you would be most familiar with is in your bone marrow. You have stem cells that give rise to your blood cells. So, uses of stem cells. Somatic stem cells are already being used to treat diseases, including Parkinson's disease and some types of cancer. Let's talk about the moral and medical advantages of using somatic stem cells rather than embryonic stem cells. Somatic stem cells can be grown without the destruction of human life that the use of embryonic stem cells involves. Also, somatic stem cells have already been used successfully against some diseases. Embryonic stem cells have not. In fact, they had high hopes for embryonic stem cells, and they thought that they would be the more useful of the two types of stem cells, and they have found that they actually are not. Somatic stem cells seem to be easier to grow than embryonic stem cells and easier to differentiate into specific cell types. Their use also eliminates the possibility of rejection if they are from the recipient's own body. Even stem cells, much like organ donation, there is that risk of rejection of these cells because the body would realize that it's not a part of it. And so the individual would have to be on immune suppressing drugs so that their body would not reject these cells. Now, as a side note, the Human Genome Project, you've probably heard of it. It was completed, the decoding of each human gene in the year 2003. Its primary activity was sequencing the human genome, just seeing what was there, and it gave us a lot of valuable information. It revealed that non-coding junk DNA is used for gene expression. When I was your age, I was taught that our DNA was mostly junk DNA that did not code for anything. In fact, only about 1.5% of your DNA actually codes for proteins like we've talked about in class, and we did the decoding. What they have found is that junk DNA that they thought did not do anything previously is used for gene expression, which is valuable knowledge. It just shows that it does have a purpose and really refutes evolution in that manner. They thought that the junk DNA was there because it was a result of evolution and it was just DNA that we no longer used. They also found that there are only about 20 to 25,000 genes in our DNA that actually codes for proteins. They thought it was going to be closer to 100,000 because of the number of different proteins that we do make. It's a lot smaller than that, but even since the Human Genome Project, one thing that they have discovered is that if the DNA code is read one way, it codes for one protein. If it's read backwards, it codes for another protein. So we're discovering new things about our amazing DNA that God created each and every day. So our objectives today were to define stem cell and explain the difference between somatic and embryonic stem cells. Don't forget to answer your 5 questions in the margin, and also don't forget to keep watching this video to answer the questions at the end.